Assalamualaikum warahmatullahi wabarakatuh, and hello everyone. Today our group will be doing a short activity called the Paper Gene Impression Challenge. The main goal of this activity is to help us understand how to identify the important parts of a gene, specifically the coding regions called the exons, the non-coding region called the introns, and the promoter region, which is found just upstream of the gene. By the end of this challenge, we will have a clearer idea of how genes are structured and why each part of it is important for proper gene function.
In this 60-base DNA sequence, the promoter region, containing the TTAA box, is located about 20 bases upstream of the start codon.
So firstly, before we start off everything about the gene functions, I'm going to explain about the most important part, which is the start codon. So firstly, before any gene function such as making protein, translation or transcription could happen, it must be started with a start codon. Most of the time, the start codon is ATG on DNA and AUG on mRNA. So basically, the start codon function when it is recognized by the ribosome, which is we know ribosome as the protein synthesis site. So basically, the ribosome use ATG or AUG to set the correct frame for every codon after it is read in the right group of 3. Other than that, it is also recruits a tRNA carrying methionine, which starts the amino acid chain itself. So it is important for the start codon for the other process of genes to be happen. Vice versa, without the start codon itself, basically ribosome will not know where to begin the protein synthesis, and plus the protein could be built incorrectly or not at all. So basically, that's for the start codon. Thank you.
And the stop codon, TAA, shows where it ends.
The stop codon is like a full stop in a sentence. It tells the cell when to stop making a protein. Our body works like a busy factory that follows special instructions written in DNA to build proteins, which make up our hair, muscles, skin, and many other parts. When the cell reads this DNA instruction, the stop codon acts as a signal that the protein is complete and no more amino acid should be added. Stop codons terminate translation, meaning they do not specify any amino acid and have no complementary tRNA or anticodon. Instead, they cause the release of a complete polypeptide chain, ending the protein making process. It's just like a recipe that says make sandwich, add cheese, and tomato, stop. The word stop means it's done. Similarly, the stop codon tells the cell that the protein is finished. There are 3 stop codons, UAA, UAG, and UGA, and all have the same important role to end translation correctly.
This is where RNA polymerase binds to begin the transcription. After that, the start codon, ATG.
The promoter is a short DNA region located just before the start of a gene. It functions as an on switch that controls when the gene should be activated. In this part of the gene sequence, the RNA polymerase enzyme and other transcription factors attach to begin transcription, which is the process of producing mRNA from DNA. Without the promoter, RNA polymerase would not know where to begin reading the DNA, and the gene would not be expressed properly. Although the promoter does not code for any protein, it plays a crucial role in regulating how much, when, and where a gene is expressed. For instance, some promoters are active only in certain cells or under specific conditions, ensuring that the right proteins are made at the right time.
For demonstration, I will divide the sequence into 2 exons and 1 introns.
Did you know that introns are actually the non-coding regions of a DNA sequence? Unlike exons, introns are not involved in the production of protein during the translation process. Introns are located in between the exons, which are the coding regions of a DNA sequence. During the RNA splicing process, introns are removed completely from the mRNA strand.
On the other hand, exons are the inverse of introns. Exons are nucleotide sequence in DNA or RNA, which comprises the genetic information for protein synthesis. During RNA splicing, introns are removed by spliceosomes, and the remaining exons are joined together, with the addition of 5' cap and 3' poly A tail to form a major mRNA. Interestingly, throughout the nucleic splicing, exons from the same gene can combine into different sequences to form different types of protein. This helps increase protein diversity despite the limited number of genes, allowing for different functions in different tissue and condition.
Well, when the stop codon, usually which is AUG, it mutates, the ribosome often fails to recognize where to begin translation. And since AUG, the triplet we have, it defines the reading frame. So basically, it's lost. It means no full-length protein or functional protein is made, and the gene's overall function is lost. And sometimes the ribosome may start at a downstream AUG, which creates a truncated protein that may misfold or localize incorrectly. And in rare cases, it can initiate a near cognate codon like CUG, but that's inefficient and it produces very little protein. And in eukaryotic cells, failed initiation can also trigger non-cis-mediated decay, which leads to the degradation of the messenger RNA. So a single change in the codon can either silence the gene or it can completely yield defective protein.
Now let's take a step back and reflect on what we have learned. To begin with, exons are the coding section of a gene. They carry the instruction to make a protein. Intron, on the other hand, are the non-coding section formed between the exons. Although introns don't code for protein, they still transcript into pre-mRNA and later removed during RNA splicing. So what's left are the exons, which join together to form mature mRNA and later translated into a protein.
I will explain more about how to distinguish them in a gene conceptually. Here shows a model of pre-mRNA, where 5' of UTR or untranslated region is located at the beginning, and 3' UTR is located at the end of it. We can see the introns, blue color regions, are interspersed between the coding exons, yellow color regions in a gene. Usually, the first exon is the start codon, AUG, while the last exon will be one of the stop codons, including UGA, UAA, and UAG. Afterwards, RNA splicing occurs, where a spliceosome will cut those interspersed introns and join all the exons together to create a mature mRNA, just like this. As you can see, all the blue color regions are cut out, and all the yellow regions are connected. In a nutshell, in a pre-mRNA produced from transcription, the non-coding regions, mainly introns, will be removed during RNA processing before translation process starts, conducting by spliceosome.
There are three possibilities of mutations in this case. Firstly, a mutation that weakens the promoter. As a result, the RNA polymerase would not be able to bind to the promoter as effectively, causing less or no mRNA made. Secondly, a mutation that completely destroys the promoter. In this case, the RNA polymerase would not be able to bind to the promoter at all, causing no gene expression. Thirdly, which is a rare case, but a mutation that increases the binding affinity of the promoter, causing overexpression of the gene.
The loss of a stop codon has no effect on the transcription, as the process ends at the terminator sequence, not the stop codon. However, it has a major impact during translation, as the ribosome does not recognize a proper termination site and continues translating beyond the normal coding region. This results in production of an extended protein chain, with often misfolds and becomes faulty and unstable. Such abnormal proteins may lose their normal functions or even become toxic to the cell.